Okay, thank you all for being here. My name is Freya Pappert. I recently graduated at Kiel University, north of Germany, and I'll be showing to you a chapter of what I did during my PhD. So hoping this works. So our group focuses on male pregnancy and the evolution of male pregnancy. And we know that viviparity has evolved independently around 150 times, but male pregnancy has only one single evolutionary origin, and from there has diversified across 300 more different species of different degrees of intensity of pregnancy, from the very simple straight-nosed pipefish, where the female just puts the eggs on the torso of the male, all the way up to pipefishes and seahorses, which have a kind of pseudoplacenta, where they actually take care of the offspring in a very vascularized pouch. Now, today I'll be talking to you in this project about the pipefish, Signatus tifler. So I'm going to show you a quicker video, which thankfully works. So what you see here is in a seagrass bed. This is the male at the front, and the female has spotted him. And these are sexual reverse species. And so the female is now kind of courting him and going on the side, and soon, hopefully, you might be able to see she will have striped bands on the side of her body, and those are sexual markings to kind of identify, you know, I'm sexy, I'm ready, like, please take my eggs. And the male, on the other hand, he will start to shake a little bit, like spasm, and that's an indication for her to know, okay, I am ready actually to take up your eggs. And yes, soon the deed will be done. And then, so here you can see she's now putting the eggs inside, and she'll fill up his pouch. And then the pouch will be sealed, and he will fertilize the eggs. And then the embryos will stay in there for roughly a month. So maybe you can see, I don't know if the lighting's great. There's like orange kind of spots on the pouch. And so it's really cool for us in our lab because we can immediately identify if they're pregnant or not without actually witnessing the mating. And then after a month, the male has been pregnant for this time, has been taking care of the offspring in his pouch, and he will then give birth to fully fledged little pipefish babies who are ready to face the challenges that the world has to offer them. So yeah, so you can see now giving birth. And there's a little pipefish, so they'll roughly like between 50 and 100 offspring will come. So yeah, and that's it. So, oh, that was it because it didn't work. So the really cool thing about working with this model species is that in conventional species, we have two really costly traits, which is egg production and pregnancy, which are both in the female. And here I'm able, in Signatus tifler, to actually uncouple these two things, so the costly egg production and the pregnancy. And this is really interesting because if we think also of transgenerational plasticity, you have environmental cues that are going to modify what's being passed on by the gametes, but also the signal cues that are happening while the mother is pregnant. But sometimes it can be a little bit difficult to really say, okay, which of the two is now really responsible for what we see in the offspring. And this is also important because the offspring wants to be prepared for the environment that it's going to be in. And this also, we've seen that if you have an environmental mismatch, which you're preparing your body and traits for an environment that is the opposite of what you expected, you can actually have diseases and not live as long. So this is really important. And so now coming back to the pipefishes, it's also similar for them. They live in the sea, and they have all kinds of environmental fluctuations from temperature, salinity, food amount, different bacteria that they may come in contact with, and pathogens. And these are definitely going to affect the gametes of the parents. And this changes over lifetime, what you're exposed to from when you're young to when you're old. And on top of that, you also have senescence happening. But as I said before, we're also interested in looking at what happens during the developmental stage, so with pregnancy, and how those environmental cues might directly already prepare the offspring. So yes, and this kind of came to my research question. So how does parental age actually affect offspring in a sexual reverse species that has male pregnancy? And, you know, and how does a young pipefish compared to an old pipefish actually affect offspring quantity, number, and quality? And so to do that, As I said, I'm from the north of Germany in Little Kiel, and we have the Baltic Sea, and this is where the pipefishes actually live in the seagrass beds. And what you do is you just go snorkeling and you catch as many pipefishes as you possibly can. You bring them back to the lab, and we have aquarium systems all prepared with the right environmental systems. And from there, we took morphological measurements of size, weight, and we looked at pouch morphology. And then we took really two diverging categories from very young, so pretty much just sexually developed, to really old, so over 2 years. So typically they don't live longer than 3 years, so 2 years they're already like pretty long and big. And then we crisscrossed all these different age categories, and then we let the pregnancy go through, and then once they gave birth, we measured morphology of the offspring, and then we also looked at gene expression of the different offspring from the different parental combinations. Now, so for the results, so first and foremost, I just have to say the pregnancy length was the same, so there was no difference. So that's just important to say that there was no developmental stage difference. And the first results that I want to show you are actually the offspring count, so how many offspring did each parental group have. And so on the x-axis, we have old males and young males, and then in the different colors, we have in blue old females, and in orangey, pinky, whatever it is, young females. And the main thing that I want to show you here is actually that there was a strong difference in the offspring number, whether a male mated with a young female or an old female. And this is really cool because it actually confirms this sex role reversal that we see, that an old male is going to be a lot more picky, and he is not going to allow a younger female that he is not considering very sexy to fill up his pouch with as many eggs as he can, whilst on the other hand, an older female, he will allow to fill up the pouch to the fullest. And we also see this in the wild, that actually males can have multiple egg batches from different females in one pouch. So that was really interesting. And then the next thing that we saw when we were looking at offspring length, again, the main thing that I want you to focus on here is actually that, again, old males and old females had the larger offspring. And this is actually what we kind of predicted. So you see this in fish with indeterminate growth, that the larger they are, also the larger the eggs are. And this tends to then correlate with offspring size at birth. And then males probably also are able to contribute a little bit more if they are older. So it seems in this case, at least morphologically speaking, that having older parents in this pipefish species seems to be quite beneficial, because if you're a young fish when you are born, you have a competitive advantage of being able to swim faster, hunt better, and be able to escape predators. Now, when we were looking actually at the results of the gene expression analysis, as I said, we were comparing offspring that maybe had a young mother and an old father, or just old parents, or just young. And so here I'm going to immediately show you both figures because they complement each other. So on the PCA, what you see from the offspring is that on the left-hand side, you kind of have just offspring that are having an old male as a father, and on the other side, you have offspring that just have a young male as a father. And so that's already interesting. So it seems that the father is the one that's kind of driving the differences. And then when we actually did the pairwise comparisons, we really saw no female effect, but a really strong male effect. And it was even the strongest, I don't know if I could show you this on the second one, when you just had an old male as a father. So there seems to be something going on, that it is the life history that is probably also affecting the offspring gene expression. And because of time and so on, I won't go into too much depth, but I'll show you the gene ontology analysis that we did when we looked at offspring that had an older male as a father. And the main thing that we find, so there was a lot of different pathways, but the general picture that we saw is that there were a lot of pathways related to cell communication, cell growth. These were some of the most dominant pathways, so MMPK pathway, which is important for signal transduction, and also the regulatory RAG complex. There were a lot of genes within and also mTOR that were upregulated. And typically, if I would see this in an older animal, I would say, okay, maybe this is a rapid onset of senescence. But because this was in young offspring, I actually think that this is just them emphasizing growth, which is what kind of you want. And then we also found a lot of pathways relating to DNA replication initiation, and cell cycle regulation and DNA repair. So there seems to be a lot of building, deconstructing that's going on in these offspring that have an older father. And we saw the opposite when they had a younger father. With the mothers, we couldn't see many results because there just weren't that many differentially expressed genes. So my take-home messages are, so first and foremost, it really did appear that offspring that had older parents were at an advantage when it came to the physiology. So again, because they were also larger, they are probably able to hunt better, so this is our idea. But the main thing that I wanted to hone into was the fact that in this case with male pregnancy, we're kind of seeing the exact opposite pattern as we would see when we would have female pregnancy, that the mother seems to have such a strong influence on the offspring. In this case, it seems to be actually the pregnant father that had such a strong influence on the offspring. Of course, at this stage, the next thing that we want to do is look at longitudinal data and see how this would affect the offspring once they grow older, because also it's hard to disentangle 100% what are actually things that are happening during the pregnancy from the environmental cues and what are life history things that the father has accumulated, which are actually giving a signal to the offspring. So yes, those were kind of our main findings. If you want to read more, you can go and read up the paper that's been published. And I wanted to thank the people that have been involved in this research and my colleagues from Kiel University and my PI, Olivia Roth. So thank you very much. I don't know what my time is. Oh, yeah.
Do you have any evidence of things that are transferred to the offspring from the male?
So yeah, so this is one of the difficult things. So there's one study that shows that there is some glucose transfer, but it's not to the same degree as you would have in seahorses. So seahorses, we see proper true vascularization, and there is nutrient transfer. In pipefish, it's a level lower. So we see a lot of osmotic regulation. There's also immunological components that are being passed on from the father to the offspring, like antibodies and so on. So there is communication, but it's not to the same level of vascularization. What we've seen is that the eggs are porous, and there seems to be some communication that's going on. But of course, when we talk about pregnancy, it's not the same degree of pregnancy you would have in mammals, of course. So yes, but there is still something being passed. But I wouldn't say it too strongly. I hope that answers. Oh, God, okay, yeah.
So your lab setup is monogamous mated, right? So
Mm-hmm.
I was kind of surprised that the old males would be picky with the young females if they don't have anyone else to reproduce.
Yes, we thought the same, but the point is, you could be that the male is just waiting and maybe thinks, okay, at another opportunity, there might be a female coming along, and I could allow that to happen. So there are other studies that have let one old male with a lot of young females, and he does mate with all of them. He doesn't go with just one, but typically if there's an older female, they tend to just mate with the one. Yes, there was both of you, but the gentleman.
Thank you. Very interesting. I was wondering some methodological things. I probably missed it.
Probably.
How do you age these?
Okay, so this is the point. So we had otolith measurements, but unfortunately they're so tiny, it didn't quite work out. So what we ended up really doing is we have just length and weight, and if we look, I don't have it here, but if you do a histogram, you can see that they are both at two extremes. So of course, in fish with indeterminate growth, you can't be 100% sure that there's not some environmental cue that might speed up growth or whatever. We did kind of try to correct for that, but it is tricky. But what we know, because we have kept these fish in the lab over years, we are pretty accurately able, thanks to all the otolith measures that we have taken in the past, to kind of date, okay, this is kind of just sexually now matured. We can see this from the pouch. And the older ones, they are just massive, and we know that those definitely have to be over 2 years. So again, for this study, we didn't have otolith measurements, but we have otolith measurements from previous studies, and those were kind of what we used to compare with size and weight.
So this individual variation in size that is much smaller than
Yes.
the age?
Yeah. So we really tried to get the two uppermost extremes because we couldn't really account for that. So yeah. Sorry, yeah.
Oh, I was wondering if you have any hypothesis as to why male pregnancy evolved in this species? Because usually you expect because the eggs are kind of larger, so maybe it conserves more energy if the female gets pregnant.
Well, yeah, I think so. We see in fish that there is a high degree of parental care generally. So that evolved kind of first. And I think male pregnancy is kind of a next step where it's just like, I don't want to have to avoid eating and having the babies in my mouth. I don't want to have to maybe be fanning them. So the next step is like, if I just have them on me or in my pouch, I can kind of just swim along and do my own thing and still kind of take care of them. And then, of course, you always have higher and higher degrees of evolution where at some point they are properly giving nutrition and taking care. So that's kind of their hypothesis. So yeah, I don't know what time it is. Is it? Okay, good. So thank you very much.
Well, thank you so much for coming to my talk also. So I'm going to present my one chapter of my recently finished PhD, which was looking at species longevity and its mediating effects on the trade-off between investment in current versus future reproduction in birds, which we did through a meta-analysis. So it falls most broadly into life history theory, hence the session, which is simply just a framework within evolutionary biology or evolutionary ecology for understanding the adaptive response of organisms to different environmental factors across their lifespan. And so simply there are genotypes that exist within a population acted upon by different selection pressures, which ultimately are expressed as different phenotypes. And these phenotypes then influence how much resource is available to individuals and how they allocate that resource to different life processes, growth, maintenance, reproduction, etc., which ultimately influence their fitness outcomes and in turn which genotypes are available in the population. And so given the kind of broad scope of life history theory, it is a very popular topic for much research. So almost 130,000 hits from kind of a rudimentary search of Web of Science, excluding irrelevant databases. So a fundamental concept of life history theory is the idea of trade-offs, that the individuals are always trading off between different processes that they're carrying out. So this is postulated to exist both within and across different aspects of life history theory. And given the abundance of available literature on the topic, in more recent years, there have been a rapid increase in number of meta-analyses being conducted. So still not too many, but increasingly meta-analyses are being conducted to kind of test whether these broad predictions of life history theory, especially relating to trade-offs, can be shown across different species. And so, for example, this study on 17 different fish species found that there was indeed increased offspring reduced offspring size whenever there was an increase in offspring number. However, some other meta-analyses, for example, this in 19 different bird species, found that while increased parental investment led to reduced adult survival in males, this was not the case in females, which kind of partially supports the existing predictions, but not fully. Or in the case of plants, this meta-analysis of 120 different plant species found that plants with a longer lifespan indeed had greater biomass, but typically had reduced fecundity or produced fewer seeds in a year. So within life history theory more broadly, there is parental investment theory, looking at these trade-offs typically in how parents invest in their offspring. And so the most typical or most commonly researched trade-off within parental investment theory is the idea that individuals only have a certain amount of resource to give, whether that be time, energy, and so on. So any resource that they invest into their current offspring is ultimately unavailable for them to invest in themselves and their future offspring. And so the question becomes, how much should they invest in that case? How much should they invest in their current offspring, depending on how many, for example, future offspring they're anticipating having? This comes down to various factors, largely related to the demand of the current offspring and similarly the value of that offspring. So for example, if an individual has more offspring, the demand of that offspring is higher, but also the value of that current brood is higher. And also environmental factors will play into what they can actually do in supporting those offspring. So things like inclement weather or prey availability will ultimately influence how much they have available to invest in their offspring and how they choose to invest that, whether in current or future offspring. And so these things taken together result in plasticity in parental investment in offspring. And so parents choose, in some sense, how much to invest in current versus future offspring. Studies looking at this typically do studies in birds, largely because the avian class is diverse in its life history strategies and behaviors exhibited by individuals. But also they have multiple breeding attempts in their lifespan, meaning they need to manage this trade-off between investment in current versus future offspring. They're also accepting of the young of others. They typically amenable to experimental manipulation. So for example, brood size manipulations, which is a common way of testing this kind of trade-off. And they typically have easily observable behaviors even in field settings. And so myself and collaborators decided to look at the existing literature on this topic through a meta-analysis. And so the predictions related to trade-offs in reproductive investment are seemingly intuitive. If you invest more in your current offspring, you ultimately have less to invest in your future offspring. There should be a negative relationship between these two. But actually, at the level of a population, whenever researchers conduct studies at a population level, they typically fail to find this expected negative correlation. Instead, often report a positive relationship between investment in current versus future offspring, meaning it seems at the level of a population that individuals which invest more in their current offspring also invest more in their future offspring. However, likely this is not the result of kind of a true lack of trade-off, but rather inappropriate study design. So this individual level trade-off is lost that you only look at a population level. So there's a kind of classic study by Van Overbeek and de Jong in which they likened this to kind of a simple example for us to understand, which is that, you know, if I or you invest more in buying a nice house and more money, then we ultimately have less money available to buy a nice car. But if you look at the level of a population, it seems that individuals with nicer houses also have nicer cars. And so it's not that they are somehow investing more from the same pot into both, but rather that they have more resource available, or in that example, more money available. And so if there are across population differences in how much resource is available to individuals, then we lose track of that within individual trade-off. And so even though if we look at the level of individual, we can see that it is lost at the level of the population. And so a great way to disentangle this in many species, especially altricial species, is through experimental manipulation of the value of the current brood or the size of the current brood. And so researchers will enlarge or reduce the size of a brood of offspring and track how the parent responds to that. And so this is what the studies that I included in the meta-analysis that we carried out. So I had two main predictions related to this, which was that parents should change their level of investment in their current brood relative to the value of that brood. So if researchers experimentally increase the size of a brood, they're also increasing the value of that brood to parents relative to expected future offspring, and so they should increase their investment in that current offspring. And oppositely, if the value or the size of the brood is reduced, parents are likely to reduce their investment in that offspring and instead reserve more energy for anticipated future offspring. But I also expected that this might be mediated by longevity. So longevity in this case simply providing a proxy for residual reproductive value. In other words, how many reproductive attempts an individual might expect still to have in its lifetime. And so longer-lived species we can expect might have a higher residual reproductive value. They're expecting to have more reproductive opportunities in their life relative to a very short-lived species. And so this led to kind of key predictions of this meta-analysis, which were that shorter-lived species, when faced with a reduced brood or an enlarged brood, are very unlikely to be able to change their behavior very much. Right? So a shorter-lived species, imagine it only has this reproductive opportunity left, or maybe one more. It's already likely to be investing as much as possible into that current brood. And so if you increase the value of that brood, it doesn't have much more to give to invest more in that brood. But if you reduce the value of that brood, you're reducing it much less than if it was a longer-lived species, right? It's still not expecting to have many more opportunities, and so the value remains quite high. It's unlikely to reduce its behavior or investment very much. But longer-lived species, when you reduce the value of their brood, you've now afforded them the opportunity to invest more in themselves and future offspring. So they're much more likely to have a greater reduction in investment in a reduced brood. And in terms of what they will do in response to an enlarged brood, they could either invest much more, given that they kind of have that energy available to invest if they're not investing as much as possible in their current brood, or they might still choose to prefer investing more in future reproduction instead of their current brood. And so we tested this using a meta-analysis, in which I'm going to skim over some details in the interest of time, but we screened, extracted, and analyzed relevant literature. That literature had to conduct an experimental brood size manipulation, either reducing or enlarging the size of brood, or many studies did both. It also needed to report some measure of parental investment, meaning, for example, how often a parent provisioned their offspring, what the size of that prey was that they were provisioning, etc. And also needed to present results that were extractable, which surprisingly ruled out several papers. But they needed to present results from which we could extract information to ultimately calculate hedges G for comparison across different studies. And so from our searches and exclusions, inclusions, we ended up with 52 different studies from which we could extract 301 estimates. So these estimates were pretty evenly divided across brood enlargements and brood reductions. But the most commonly reported response variables were feeding rate, which had over half of the estimates, and also nest visit rate, which is typically used as a proxy for feeding rate in some species. Our studies came from 17 different countries, covering 5 continents. However, maybe unsurprisingly, it should be noted that most of these came from North America and Europe. So we had also 26 different species included in our meta-analysis. On the left side here is the phylogenetic tree of included species. On the right side is the average number of breeding years as our proxy for residual reproductive value. We had 26 different species included, but again, 45% of these came from 2 species, great tits and pied flycatchers, which again, probably not so surprisingly if you're in the field of bird research. And we also had a variety of average breeding years, some short-lived, some longer-lived species. The shortest being the red-winged blackbird, which had less than 1 average breeding year, and the longest, the brown pelican, which had around 17 average breeding years. And so we included and tested various different models with different moderators, categorical and continuous, but for the purposes of this talk, I'm just going to talk about a few of those, meaning the treatment type, the relative change in brood size, which is simply the percentage change in brood size relative to the control, the average lifespan, and also the average breeding years, or the average lifespan minus the average age at first reproduction. And so what we find is, in terms of our first prediction, which was that there should be a change in investment with brood value, in interpreting the graph here, the dotted line represents no change. So I coded all of my estimates to be positive if they were in the expected direction according to our predictions, meaning a positive change means one in the direction we expected it to be. And so anything above this line means that that was a change in predicted direction. And so what we find is that for both enlarged and reduced treatments, parents significantly changed their level of investment. So they significantly increased their investment in enlarged birds and significantly reduced their investment in reduced birds. But as you can see from this figure, there was really not too much of a difference between how much they changed their behavior for enlarged versus reduced birds, which is somewhat unexpected. And so for each of my models, I also calculated the marginal R-squared value, which simply tells us how much of the variation in the data was explained by the moderator included. And so when we included only treatment as a moderator, actually we find that it in itself explained only a very small amount of the variation in our data set. But there was a much stronger relationship or change in behavior relative to the change in brood size. So the greater the amount that the brood size was changed by, the greater the response by parents. And this too was linked to their whether it was an enlarged treatment or a reduced treatment. There was an interaction effect between those two, such that including both treatment and the interaction effect between treatment and relative change in brood size explained a large amount of the variation in our data. In other words, parents responded most strongly to a very reduced brood and least strongly to a smallly increased brood. So in regards to our second prediction, however, which was that this should be mediated by species longevity as a proxy for residual reproductive value, actually we did not find evidence of this. So we find that including treatment and the interaction between treatment and average breeding years actually explained only a small amount of the variation in our data, and there was no strong relationships between average breeding years and change in parental investment. However, as you might have already noticed from these figures, the available literature is very skewed in what age the lifespan of species included are. We had almost no studies which conducted research in bird species which lived long, meaning they had an average breeding years of over 5. And so to kind of bring that all together, conclude, we did find that across bird species, parents adjust parental investment relative to the value of their brood. And so this provides a support for really key tenets of life history theory, right? The parents should adjust their level of investment in current versus future offspring. But we did not find, based on currently available literature, that this was mediated by longevity as a proxy for residual reproductive value. And so I kind of leave you with the thought of whether you think this is a kind of real instance, really it's really not mediated by longevity, or whether this is simply the result of the kind of data gap in existing or available literature. And simply if we had more longer-lived species, studies in more longer-lived species, we might find the relationship. And so this is part of my PhD thesis, and so I have various funders to thank, as well as my PhD supervisor, Dr. Kimberly Matteau, and various others who have collaborated on the project. If you have any questions about other things we did in this paper, there are many more I, of course, couldn't include here, or anything else to do with the research, you're welcome to ask now. We have 30 seconds, sorry. Or you're welcome to contact me on LinkedIn via this code, or I'll be around. Thank you so much for this. Sure, go ahead.
Did you consider other factors that could mediate this trade-off? Because I'm thinking, for example, for ecological factors like resource availability in a particular year, that could mediate this trade-off between the long-term versus short-term phases components investment on short-term loops, long-lived species. What I read is like in short and long-lived species you can have particularly the season has to skip reading in a particular year depending on this, which
Right.
cannot because they don't have too much opportunity to.
Makes sense. So we were kind of limited on what we could include in that it's a meta-analysis, right? So we can only extract data that exists in that has already been published. And so we extracted kind of everything from every possible paper. And I originally had the idea of if a study reports things like different years, then we extracted those separately, right? And we originally intended to kind of try to do some sort of analysis where we looked at different years and things like that. Or if a study reported prey availability on the landscape or other things that we expected might influence how much a parent might invest other than just the demand of their offspring. But there was not enough, essentially, from what already exists for us to be able to do a kind of solid analysis using that. But I'm happy to talk kind of anecdotally about what was in there really after. Sorry, I'm invading someone else's talk right now, but thank you so much for your question.
So I'm Carol Boggs. I'm at the University of South Carolina and also work at Rocky Mountain Biological Lab. And I'm going to talk today about the fitness consequences of variable food stress. There's lots and lots and lots of studies looking at the effects of continuous food stress throughout a lifetime for quantitative or qualitative food stress on all kinds of life history traits, on metabolism, on behavior, etc. There's also a lot of selection experiments that have been done involving lifetime food stress. And this has been done largely in the context of looking at phenotypic or genetic trade-offs, looking at the evolution of senescence, looking at the role of macromolecules in life history trait expression, and so on. But the problem is that in reality, stress is not constant for the most part. So you may have weather events that come in on a short-term basis relative to the lifespan of the organism. You may have climate change happening. You may have an organism such as a cow that comes in and eats all of your nectar plants. There's a variety of different things that can happen that can alter the availability of food only over part of a lifespan. So that's what we wanted to focus on at this point. So the study organism that I've been working with for many years is the Mormon fritillary, Spirea mormonia, known on iNaturalist as Argynnis mormonia. This is in the nymphalidae. It exists in montane North America. The habitat is shown here at Rocky Mountain Biological Lab, and the beastie is up there. The females as adults nectar on members of the compositae, and this means they have an incomplete adult diet. So they're getting primarily carbohydrates in the adult stage, and most of the nitrogen has to come from larval feeding. The eggs that the females lay have a high carbon to nitrogen ratio, in part to support overwinter survival because the egg's diapause is unfed first instar larvae. And about 80% of that adult carbon in the egg at equilibrium comes from the adult diet, based on work that I've done with Diane O'Brien. So we also have, usefully, background information showing that quantitative food stress or adult dietary restriction across the entire lifespan results in the preservation of lifespan at the expense of fecundity. So if you feed a female 1/2 of what she would normally get, she lays half as many eggs. And furthermore, this translates into the field in terms of the effects on the population dynamics. So we know that there's significant serious fitness costs to quantitative variation across the whole lifespan, but what happens in a shorter time period? So we had to think about how we were going to define this shorter lifespan. And this is a diagram of the days of the female, the age of the females against the microliters of the adult diet. And as you can see, they're really pigging out in the first 3 days or so. So a huge amount of nectar coming in is coming in the first 3 to 5 days. So we want to focus on that because that's where we think we're going to potentially see the biggest effect. It's also when they're doing a lot of egg provisioning in this case. So our hypotheses are that early adult food stress versus ad libitum, or eating as much as they want, food, is going to depend upon, the effects are going to depend upon whether or not there's compensatory feeding after that food stress is done. So if there's compensatory feeding, they're getting as much nectar or sugar water or whatever you're feeding them as they would normally eat, and we expect the fecundity will be unaltered. Lifespan might be increased if they need a little extra time to get all of those nutrients, but it may also be the same. If there's no compensatory feeding, we expect fecundity will be decreased, although not by quite as much as it would be if they were continuously stressed over the entire lifespan, and we expect lifespan to be constant. We added on another experiment because it's not only that a cow can come in and eat the flowers, but you can get monsoonal rain coming in from through Arizona, in which case it's cold, it's dark, and it's wet, and no butterfly in its right mind is flying anywhere to do anything. They might as well have been thrown in a refrigerator. So we tried cold stress. We threw them in the refrigerator to see whether or not that had the same effect as food stress. So the methods, we caught freshly eclosed and mated females in the field. This was done in the summer of 1993 and 1994, well before most of you in this room were born. So this is digging back into old data. And we had adult feeding treatments. So we had controls where we measured the wing length, which gives us an estimate of the body reserves from larval feeding, so the estimate of the body size. And the first member of the pairs got as much nectar as they wanted to eat, or as much honey water, rather, as they wanted to eat 2 times a day. And to measure that, we used Hamilton syringes. We uncurled the proboscis with the needle and then measured with a Hamilton syringe how much they were going to eat. Pretty labor-intensive. The second member of the pair got half of the 2-day average of the ad lib fed female. The treatments, the third pair member, got half ad lib for the first 5 days and then got ad lib for the rest of its life. And the fourth member here went to the fridge for either 3 or 5 days, so this is 4 and 5, and then got ad lib after that. And we recorded wing length, we recorded the microliters that they ate, we recorded their lifespan, and we recorded the daily number of eggs. Okay, so the first question is there compensatory feeding? And the answer is definitely yes. On the x-axis is ad lib, half ad lib, so the 2 controls, and early stress. 5 days, half ad lib, and then ad lib, and total microliters eaten across the top. The treatment is significant. The post hoc tests show that there's no difference between ad lib and early stress. And as you would expect, given the experimental design, half ad lib is significantly less. Okay, so this means from our hypotheses, we're into this left-hand side here. Compensatory feeding, we should expect to see fecundity unaltered, maybe lifespan increased, but maybe not. We'll see what happens here. So if we look at fecundity, in this case, What we see in terms of the number of eggs that they lay, treatment again is significant, and there's no difference between ad lib and early stress, and a significant difference with half ad lib. So we've replicated all our early studies with the half ad lib one, but there's no significant difference with post hoc test, which says that fecundity is indeed preserved. The hypothesis is right. If we look at lifespan, there's no significant difference across any of these treatments in terms of lifespan. Lifespan had a probability of 0.2. If you kind of squint, it looks like it might have gone up for the half ad lib and the early stress, but that's not a significant effect. So if we go back to our hypotheses, there is compensatory feeding, fecundity is unaltered, and fecundity, yes, unaltered, and lifespan is not increased. It basically stays the same. So what happens if you throw these things in the fridge? What happens if you have cold stress? We first need to look again at compensatory feeding. And here we've got total microliters again along the y-axis. In this case, the ad lib fed ones first, then 3 days in the cold, then 5 days in the cold, then our other control, half ad lib, and then we're hanging in here with the 5-day food stress for comparative purposes. Treatment is significant. There's no difference between the ad lib ones and the 3 days in the cold, and then no difference between, in post hoc tests, between any of the short-term food stress organisms. Okay, if we look then at the number of eggs that were laid and the lifespan, in this case what we see is there's no difference across any of our treatments compared to the ad lib for number of eggs laid. And if we look at lifespan, it's just plain not significant in this case. Well, there's an interesting thing here is that we've got sample sizes that are on the order of 10 to 12 females in each one of these categories. So our sample sizes are a little small for lifespan. And one of the things we could do, since we're not seeing any significant difference, is we can pool the data for all of the short-term stress options and see whether or not there's a lifespan effect there. And what we indeed find is that for number of eggs laid, for ad lib versus early stress, it's not significant. For lifespan, probability is 0.03 for ad lib versus early stress. So it looks like there's likely an effect of an extended lifespan in this case, presumably in order to be able to get all those extra nutrients to make up for when you didn't get them. So in conclusion, these guys exhibit compensatory feeding after early life nutritional stress. Bad weather and lack of nectar availability have the same effects or similar effects. It may take a little bit longer, but the reproductive output is unaffected by early nutritional stress. And in the wild, however, that taking a little while may make a big difference to fitness because you may end up being hit over the head with a hailstone, you may end up being eaten by a bird, you may end up with other kinds of mortality issues that would shorten your survival, and therefore, if you really needed to catch up, it would shorten your reproductive output as well. So the lab data suggest that there's no fitness effects, but it remains to be seen in the wild whether or not that's actually true. So I want to say thank you, funding to the Stanford University Field Studies Program, back when I was still at Stanford in those days, and special thanks to a whole slew of undergraduate field assistants who fed a lot of butterflies using Hamilton syringes without complaining. This is my email address if there are questions. I suspect we're running behind. I don't have a clock, but okay. If there's any time for a short question, I'm happy to answer it. Okay. Yeah.
I think I quite imagine the conditions in the fridge are like. So I assume it's dark and cold and they have limited space. Like, are there?
They're in glassine envelopes, so they might as well be roosting. So it's dark, it's cold, and refrigerators tend to be a little on the humid side, so it pretty much mimics what's happening.
It'd be interesting to see kind of what amongst those things seems to affect the most.
I suspect the thing that's happening is that the cold is keeping metabolism down, and that's making up for not being able to eat as much. Okay, thank you. Okay. Come on up.
I believe I can start. Thank you for coming. So I'm Rabi Sankar Pal. I'm from Indian Institute of Science, Education and Research. So this talk will focus on how selection for early life history alters the reproductive investment and whether it can lead to differential cost of reproduction across the sexes. So classical life history theory suggests organisms invest in different life history traits such a way that maximize their lifetime reproductive success. However, resources are finite in nature, so organisms need to actually competitively allocate these resources to different traits. This causes the trade-off between these traits. I'll focus on two such trade-offs that describe the cost of reproduction. So first is the negative correlation between the current reproduction and survival. The second being the trade-off between current and future reproduction. So before I proceed, I will like to introduce you the laboratory adapted populations of Drosophila melanogaster. So COs are the ancestral population. You can call them the control population. They are maintained in 28-day discrete generation cycle in constant environment, particularly in 25 degrees Celsius temperature and continuous light in standard banana, barley, jaggery food. The collection is done on day 0. So by day 12, almost all the flies complete their development. That is when they are transferred to the population cages. On day 25, they are given yeast supplement to boost their fecundity for 48 hours, followed by the oviposition window of 18 hours. So eggs laid during this 18 hours are collected to start the next generation. So this window very much defines their Darwinian fitness. ACOs are the evolved population. ACOs are the accelerated COs. They are selected for the faster development. Instead of completing development in 12 days, they complete their development by day 8, and they are given 24 hours of oviposition window. So you might have noticed that COs can actually have a pretty long interaction and reproductive window of approximately 16 days or so. However, ACOs not only develop faster, they need to reproduce early, and they have only effective adult life of 24 to 36 days. So all the experiments that we have carried out was done after almost like more than 1,400 generations of experimental evolution. And selection for faster development has caused a huge diversity, divergent traits. So besides their shorter development time, they are also smaller in their body size. ACO females are less fecund. They have lower starvation resistance as well as longevity. So our lab has been investigating the sexually selected traits for some time, especially those that lead to sexually antagonistic interactions. Sexual antagonism can appear when the evolutionary interest of the males and females doesn't align. One sex may invest in traits that increase their own reproductive success, even if that comes at the fitness cost of their reproductive partner. So selection for faster development has also caused divergence in the sexually antagonistic traits. Previously, our lab has shown that ACO males are less harming, although they court the females more. So females held with ACO males had significantly high reproductive rate as well as survival compared to the females that were held with CO males. So ACOs are already resource constrained. So we wondered whether reduced antagonistic effects of ACO males has changed their reproductive investment. This is particularly important because this might facilitate trade-off that can ultimately determine the adaptive evolution. So we measured the different components of the male reproductive system in order to assess the divergence in reproductive investment. So this beautiful picture from McCulloch et al. Shows the reproductive system of Drosophila. Sperm are produced in testes, and a mature sperm can be stored within the seminal vesicle, together with the secretions from accessory glands, ejaculatory duct, and ejaculatory bulb. They can be transferred to the bursa copulatrix of the female. Thereon, the sperm can be stored within the seminal receptacle for short-term or within the spermatheca for long-term usage. The sperm can be further used for fertilizing the mature oocytes coming from a pair of ovaries. So we measured the testis length. Sperm competition is known to shape the evolution of testis size. Larger testis size is known to produce higher number of sperm. So what we have seen in ACOs is that the absolute value of the testis length have gone down by almost 19%. Note here, ACOs are smaller in their body size. So we have also taken into consideration their body size, their thorax length, which is the proxy of body size in Drosophila melanogaster. So even after taking consideration for their smaller body size, their testis length was significantly shorter. We have also measured the sperm length, which was again shorter compared to the COs. So this is actually surprising because evolution of sperm length is not generally connected to the life history evolution. However, the quality of sperm is generally dependent on the length of the sperm. Larger sperm are more competitive. In ACO kind of system, due to their selection regime, the post-copulatory sexual selection might have gone down. So sexually selected traits might have been weakened. So if the sperm length and testis length are primarily determined by the post-copulatory sexual selection, then this reduction in testis and sperm length are not really surprising. We have also measured the accessory gland size. Accessory gland products are directly linked to the male fitness. And individuals having larger accessory glands are more able to produce these products. And surprise, we did not find any significant difference in accessory gland size. So although the absolute value of accessory gland reduced by 3.2%, it was not statistically significant when you consider for the smaller body size of the ACOs. Even more surprisingly, we took few accessory gland protein genes whose products are known to have a variety of functions, starting from oogenesis to ovulation to sperm defense and sperm offense. So we did not find any difference in their fold change when we compared between ACOs and COs. So accessory gland size did not reduce. And if we consider these ACP genes are a random sample out of hell lot of them, so their activity has probably also not changed. So what is happening exactly in their accessory gland? So we propose that ACO males need to be fertile very quickly after their eclosion. And many of these accessory gland products are responsible for the male fertility as well. So there might be some residual selection for these accessory gland size as well as functionality. So overall, we have seen although accessory gland size has not gone down, testis and sperm length have gone down. So this putatively indicates lower reproductive investment in the evolved populations. And not only in males, we have seen reproductive investment to have gone down in females as well. We have checked the ovariole number. Ovarioles are the functional unit of ovaries. Having higher number of ovarioles are associated with having higher potential fecundity. However, we already know ACOs have lower fecundity. This partially explains the physiological aspect of it. Seminal receptacle length is known to have a co-evolutionary relationship with the sperm length. Previously, we have seen in ACO, sperm length have gone down. Here we see the seminal receptacle length has also gone down. So we have seen overall in both males and females, the reproductive investment has gone down. So that should be reflected in the cost of reproduction. So we asked whether the cost of reproduction evolved in these selected populations. And we find a very interesting trend in the form of three-way interaction between sex, regime, and mating status. So if you can follow this plot, in the control population, you see significant cost of reproduction by means of decline in longevity in reproducing versus non-reproducing individuals. This pattern existed even in the evolved females, but the cost of reproduction in the evolved males was non-existent. So what we can interpret is that as a result of selection for faster development and early reproduction, somehow the cost of reproduction has been ameliorated in the males, however not in the females. So I'll leave this here for your interpretation. We are still looking for why this cost has been ameliorated in males, but not in females. So I'll just summarize. In males, we have seen testis and sperm length have reduced, but not the accessory gland size. In females, we have also seen the ovarial number and the seminal receptacle length has gone down. This signals reduced reproductive investment in the evolved population. However, we have seen reduction in the cost of reproduction in the males of the evolved population, but not in females. So I'd like to acknowledge my supervisor, Dr. Bodhisattva Nondi, and all my lab mates for their help in their experiments. I'd like to thank the organizers for providing this platform to share my research. And I'd like to sincerely acknowledge this Ignite Life Science Foundation for providing me the travel grant and Aizar Bodhampur for my fellowships. Thank you. Thank you for your patience. I'd be happy to answer questions.
Thank you for your really nice talk.
Thank you.
In your aging experiments, did you control the number of matings so that they only mate once, or was it just multiple?
No, so these flies were collected just after eclosion, and they were held in single-sex vials in number of 10. They were allowed 24 to 36 hours for attaining reproductive maturity in the sense their physiological maturity. And then they were frozen and then dissected to assess the reproductive system.
But in your aging experiments, when you showed the longevity, they were mated in that short time.
No, so they were either mating or not mating. So either virgin or mated. They were kept within a vial in the number of 10 single sex or 10 males or 10 females. Yes.
Very interesting talk.
Thank you.
For your accessory gland, your accessory gland experiment, what time point were those done at? How old were the flies when you measured the expression or the size of the accessory gland?
So this was done after post-eclosion. We have allowed them around 24 to 36 hours. Then we have frozen and dissected the tissues. We have followed the same protocol for the gene expression assay.
I was wondering, but for the COs, since for them mating doesn't really matter that early on, what do you think we'd see if you had waited 2 more weeks to do those experiments?
That's a good point, but we don't generally actually know whether the accessory gland size change over their regime. So if accessory gland size can change over their reproductive period, that could be a possibility. But what I have done is once they have reached the sexual maturity, that is the point I have taken.
Thank you.
Thank you.